All right, well, good evening and welcome. My name is Drew Desker. I'm the director here at Kopernik Observatory and Science Center. It's great to have you all here with us this evening on a brisk evening. I think it's going to take some time to get the blood to thicken up again, but we're upstate New Yorkers. It'll come. It's great. Well, it's great to have a nice crowd here for our talk. Just as a matter of interest, who's here for the first time or hasn't been here since your fourth grade field trip? All right, a few people. How about, who here are members? Oh, okay, good portion of the rest. Excellent. Well, great to have you here. In particular, for those people that raised their hand first, I'll point out that the people that raised their hand second have a particularly important piece of knowledge, and that's the value of a Kopernik membership. We are a nonprofit. We are not funded by any other place, so we basically are kept kept going by programs like this, our school programs, our summer camp programs, and so a Kopernik membership would get you into any Friday night program for free, but also we belong to what's called the ASTC, Association of Science and Technology Centers, and it's a consortium of over 380 other science centers. So if you like this kind of place, your Kopernik membership gets you into those other places for free. So you could go to the Roberson Museum in Binghamton. You could go to the Museum of Science and Technology in Syracuse, the Franklin Institute down in Philly, the Intrepid down in New York City. In fact, very often, especially in some of those larger venues, the cost for a family of 4 to get in would be more than definitely more than a full year's membership. So if you like this kind of place, consider getting a Kopernik membership. So up on the screen here, we've got our website. We always keep that updated with what's going on. So tonight's program is about CRISPR. We'll get into that in a little bit. But then we go down here. Vigilia 2024. People are looking at what is that? So the Kopernik Observatory and Science Center was started by a group of Polish immigrants, people of Polish heritage, back in the 1970s that wanted to commemorate Kopernik's 500th birthday. And so part of our bylaws actually is to, along with operating a public observatory and STEM education center, is to promote Polish heritage. And Vigilia is actually the Christmas Eve dinner that the people in Poland celebrate. And so actually, both of my parents were born in Poland, and so that was what I was brought up with. And so we have a traditional Polish Christmas, and you don't have to be Polish to come to it. It's a great time, great food, great camaraderie, and so if you're interested learning more about that, click on the website. But if we go over here to go to calendar of events, public programs. Here's where you find out what's coming up next. And although I need to update it. Next week, our talk is actually by a few of the professors out of Binghamton University talking about biofilms. And then the weekend after that is our standard black holes on Black Friday. That's the day after Thanksgiving. So check our website, and you'll always find out what's going on. One of the things, again, I mentioned earlier that we do school programs. We just earlier this week had our Girl Power program. We're trying to get more women involved in STEM, considering STEM careers. And we always have a female subject matter expert talking about the work that she does. And I like telling this story in that a few years ago, look, we always have a female subject matter expert talking about the work she does.
You'll be seeing some of that in my talk.
Oh, there you go. And so a few years ago, we had an astrophysicist from NASA talking about the work she does using satellite data to look at carbon dioxide levels, in the atmosphere, and how does that translate into changes in climate. And because it's a video conference, our girls get to ask her questions. And so when it was our turn to ask questions, one of the girls asked, "How did you get interested in astrophysics?" Now this is like a 3rd to 8th grader. She had a great response to that. She said, "Well, you know what? When I was in college, I was an English major, but my boyfriend was a physics major. And we'd go from observatory to observatory. I eventually dumped the boyfriend, but I kept the astronomy." Now she has her PhD in astrophysics and works at NASA. So you never know where a seed's going to get planted, and that's really sort of what we do. Throughout the year, we have a very extensive summer camp program. This past summer, we had an opportunity to have our students ask a question through the ham radio station we have here of an astronaut on the International Space Station. So as they were flying by, our students used the ham radio station here to talk to the ham radio station on the ISS, and we got to ask questions, and they answered them right back. So we do some pretty fun things here. This year we are actually celebrating our 50th anniversary. Construction started in 1973. It opened in June of 1974. And Nicholas, hold on. This fell off.
Oh, okay. Maybe I would give you a hard time.
There you go. Okay. And so about a month ago or so, we had our 50th anniversary gala, and we'll be announcing very soon a program we need to expand our facility, expand our outreach, and it's our Reach for the Stars campaign. So you'll be look for that soon. We'll be looking for some support and some guidance, and but we're really looking forward to continuing the work that we do here. So as I mentioned, every Friday night we do a public program of some sort. It's not always about astronomy. In fact, tonight's not about astronomy. And back in 2020, we had to stop doing it because COVID, right? So we ended up saying, okay, fine, we'll just hold off a couple of weeks until this COVID thing blows over, and didn't exactly play out that way for us. So after about a month or so, we said, look, we got all these speakers lined up. Why don't we do something about that? So we had them all Zoom into our facility here. We then turned that content around and put it right back out on our YouTube channel. And so if you go to YouTube, actually, if I go back here to home, I go to my videos. Yeah, this is here we go. So every Friday night program is on our YouTube channel. In fact, there are people watching tonight. So actually, everybody say hello to those on the live stream. Say hello.
Hello. Hello.
There we go. All right. And so people are literally watching from around the world. There's very often Lindsay from Adelaide, Australia, is actually watching it. For her, it's she's eating breakfast now in Adelaide, Australia, while we are talking here. So and what's also nice about these programs is that they continue to stay on our website. So if you want to go up and watch that program again, or if you missed it and you want to go and check it out, just go to our YouTube channel. We have almost 14,000 subscribers. But along with our Friday night programs, we've also done live observing sessions of lunar eclipses, solar eclipses, meteor showers. So it's a great resource for you. So as I mentioned, Friday nights are really a lot of fun. It's an opportunity for us to really continue lifelong learning. And for those youngsters in the crowd, your school doesn't end at 12th grade or at the end of college. You want to continue through your entire life. And we're fortunate to have some great speakers here, one of which tonight is Dr. Nicholas Guydosh, as an educator. He's been with us for a very long time. I won't say how long. But tonight his talk is going to be about CRISPR. And so I'm going to turn this over to his presentation here, if I get that. There we go. All right. And for those watching on the live stream, if you have any questions, put them in the chat, and then when it gets to the Q&A portion, we'll convey those to Dr. Guydosh. So, Dr. Guydosh, it's up to you.
Thank you.
Twice. And just give, just.
Okay.
There we go.
I don't hear anything.
Yep, keep talking.
Okay.
No. Well,
Oh, wait. Where's the microphone?
there it is.
Okay.
Down here.
I thought I was on it. I told you I'm high tech.
Let's try this.
Okay,
Give me a second here.
sure.
Try that.
Okay, could you hear me now? Okay. And this thing here, I keep tripping over. I will maybe put it over here.
All right, I'll just
Or maybe I'll put it in my pocket. Hey, good idea.
This way, if you start walking around, then
Yeah.
here's the.
Okay. Thank you, Drew. Ladies and gentlemen. This is not my field. My background is in electrical engineering, and I got fascinated with this thing. I guess if I could start all over again, I might want to go into biology. But I am following the advice that I gave some of my students at Binghamton University. I worked at IBM for a long time, and then I taught in the computer science department, and I always told them that the book you're using now will be obsolete in 2 years. And what I'm saying to you now and what we're learning will be obsolete. So you better learn how to teach yourself. And so that's what I do. And I still do it now. And don't ask how old I am. But anyway, you're probably wondering what that is up there. Well, I'm going to let it up there for a moment. I got into this subject by reading a book. The book, my son is into this field. He works at NIH, and once I grab my cane, I'll pick it up. Oh, also, this is my rocket fuel, Diet Coke. But it was The Code Breakers. I'd highly recommend this book if you're interested in this subject, by Walter Isaacson. It's sort of a very lively biography of Jennifer Doudna, who was the great pioneer in his field. And I'll get a little bit more into this in a talk. And I read the book and got really fascinated with it. And so I thought I'd better learn something about how cells work. So I got myself this book, and I didn't weigh it, but I think it weighs about 10, 15 pounds. It's called The Cell, and it has everything you'd ever want to know about cells in it, DNA and everything else, and stuff that you don't want to know, too. So I'll just drop it here. And I had to struggle to get this thing in. Okay. I think I should get, Sasha, you're here, aren't you? I learned that, what school were you talking about when you talked about the experiment? SUNY Broome? Yeah, maybe I should have you tell them about it, but they are doing some experiments in the area of CRISPR there. I'm getting my Don't tell Drew.
I want you to be able to stay vertical. There you go.
Okay, stay vertical. Okay, so you're wondering why the warning is up there. I just picked this idea up yesterday, and let's see. Okay, here we go. This presentation contains materials from molecular biology, which may cause anxiety. I'm starting to feel it myself. Even heartbreak. We don't want to leave you with heartbreak, okay? Because when you find out what this stuff could do, you might end up getting heartbreak. So please see your biologist if these symptoms persist after we're done here. So I thought I'd give you guys a warning on this. And without tripping over anything, I'm going to move right over to here. And I'd like to get started. And that's my title slide. Could we possibly dim one of the lights that's shining on the screen? Okay. So this is my title slide, and it's supposed to tell you what we're going to be talking about today. And I'll put the words up there now. Clustered. Regularly. Interspaced. Short. This is a cool one coming up. Palindromic. Next one, if I could. Repeats. All that put together, they didn't want to use that title, so they said CRISPR. So that's where the word comes from. And I called it the key to life's basic mysteries. And that's me. My date on the bottom, I always put a little 1 after an update. That 1 was just this afternoon. So, okay, so here's the book I told you about. This book was about the highlights of the research that's leading to the revolutionary, and it is revolutionary, CRISPR gene editing technology. We're changing our DNA with this stuff. And it was based on his book, The Code Breakers. I picked it up and showed it to you. And and the research by Jennifer Doudna. You notice Jennifer Doudna's last three letters of her name is DNA. I don't know whether that had anything to do with it or not. And Emmanuel Charpentier, who received, they both got the Nobel Prize. When Drew mentioned getting more girls and girl power involved in science, I says, some of this might come out in a talk, and this is it here. These are two women who got the Nobel Prize. So the key players, and we'll talk about them a moment here. That's Jennifer Doudna. In 2008, she did much of her work, and she's a Nobel laureate. She works at the University of California in Berkeley, and is the leading biologist in the work on CRISPR. And she received, in 2020, she got the Nobel Prize in chemistry for developing the CRISPR method for genome editing our DNA. Now, the other person is Emmanuelle Charpentier, and she joined Jennifer in 2011, and she's also a Nobel laureate. I think they both got their Nobel Prizes at the same time. And she's a French professor, you could tell by her name, I think, and a researcher in microbiology, genetics, and biochemistry. And Charpentier was awarded the Nobel Prize in chemistry for her, again, work in developing the CRISPR method along with, so they're sort of partners, they work together. I found out in reading the book, there's a tremendous amount of competition in this stuff among researchers trying to get papers out early so that they can get credit for certain ideas. And you get out too early and there might be problems with them too, you know, so you got to be very careful with this. Okay, for all life on Earth, I underline on Earth. That's all we know about so far. DNA, it's a complex molecule. It's contained in every living cell of every living thing. And it contains and implements information which describes every detail of the living thing. And I said here, including viruses. And the question I throw out, and I don't know what your feelings are on it, is a virus alive? I asked that question to some people that are into this research. In fact, my son, who is a researcher at NIH in RNA research, and it varies from, some say they're living things, some say they're just molecules that do their thing, but they do have DNA, so that says something. The big question is, should we expect DNA or a similar molecule to exist for alien life beyond the Earth? Would it, if we discovered life on Mars, or say life on a planet around another star, do you think it would contain something like DNA? Or would it be, instead of having a duplex type of a spiral, it would have three levels or something like that? No, that's an interesting question, I think. So here's something that boggled my mind. DNA, what is its size? If the DNA double helix in a human cell, just one cell, not the whole body, it could be, if it were laid out end to end, stretched out, it would be about a little over between 6 and 7 feet. And that's packed into this little tiny cell. If all the DNA in a human body, out of every cell in our body, were stretched out end to end, it would go to the moon and back 150,000 times. You could imagine how many cells we have in our body, right? That's not my idea. I read it. Okay, the nucleus, where in higher level living things like humans, dogs, cats, even a paramecium that lives in pond water, the nucleus is only 6 microns in diameter. And this is equivalent to packing 24 miles of fine wire in a tennis ball. And a micron is a millionth of a meter, which translates into 39 millionths of an inch. So that's how tiny the nucleus is. the human genome has about 3.2 billion rungs in the ladder of DNA. They're called nucleotides. That's some vocabulary that I'll throw in. And I call it the DNA ladder. And hey, wait, this. Hey, Joe, you said I could use a laser on this, right?
No, that's the big button.
Oh, the big button in the middle. Yeah, the live stream won't see this, but I just put this here, 24 miles of DNA packed into that tennis ball. Okay, I can't find the button. Here it is. So here's just a little picture of what we're talking about. This is probably a typical cell in a higher animal. It has a nucleus, a cell membrane, and there's all the DNA packed into that cell. And now I'm going to introduce an amazing character that looks like it lives in a cell. And I was fascinated with this thing because it looks like it's actually a separate living thing. And I'm going to go for it right now. And it's called a kinesin. And you can see it's sort of marching across a tubule, carrying something on its shoulders. Now this is in almost at the atomic level. And let's see, if I want to start this up, if I hit, oh, I need the mouse for this. Yeah, if you get the mouse, there you go. This is produced at Harvard. There was another version of this explaining the detail of what's happening here, and I thought I'd save that for a future talk. That would cover the whole talk. But I'm just trying to get your aesthetics up of what's happening inside of a cell. And we will see Mr. Kinesin soon. Those are tubules that the kinesin will be using. And here he comes. I say he, I don't know if that's a he or a she, or maybe it's nothing. It's carrying proteins to where they're needed in another part of a cell. That's a ribosome molecule scanning RNA, making proteins. There's a lot of computer stuff in this. And here's Mr. Kinesin now finishing its task. Okay, delivery is done, and maybe we should all give Mr. Kinesin, or it, a hand. Thank you very much. Thank you very much. Okay, I thought since we're going to be talking a lot about DNA, RNA, we should take a little peek of it. I know a lot of people probably already covered it in schools, but here's a little bit of a review. But the base, on the left we're seeing a strand of DNA. The outside thing that's holding everything together is actually it's composed of molecules of phosphate and sugars. And the ladder work in the middle of it is their base pairs. They're called nucleotides, and that's where the encoding is done. So it has its own code, and they have crazy names like thymine, adenine, cytosine, and guanine. And we make, for short, we're just going to call them ACGT. And those are, now in an ordinary digital computer, we're using binary, 0 and 1. Our own number system that we use, that we're familiar with when we go shopping, is base 10. 0 to 9 are the digits. This is going to be base 4, 0 to 3. And we'll be talking about RNA too. And RNA is, it resembles 1/2 of a DNA molecule. We're seeing it on the left there, and there's a process inside the cell that spins off RNA from DNA. And RNA is created from the DNA that has having the same information that the DNA has, but there's one major difference. DNA is just it's just a memory that stores information. Doesn't do anything with it, it just stores it. But RNA does the work of the cell function. That's the stuff that actually a ribosome is going to be producing proteins. Proteins are the main chemical or element that's produced in cells. So proteins are created using the ribosome molecule that reads the RNA molecule as it goes through. One of the differences that you may notice that in a DNA, the 4 letters are A, C, G, and T, but the T is replaced by a U in RNA, and U stands for uracil. But the function and the chemical reactions are essentially the same. Now, here's a quick review of CRISPR, and it's a video, and I'll start it up here. I got it right here.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses.
Think about this.
When the
Our virus is
bacteria
alive.
detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Could I X this out? Should I just X this out here? Okay, yeah. Okay. What else did we lose it? Okay, there we go. I got it. So I thought that was sort of a cool overview of what happens. And I'd like to get into a little more detail on it, and that's what I'll do right now. And it all started with sick bacteria. I mean, does anybody feel sorry for sick bacteria? Well, that's what happens when viruses attack a bacteria. So viruses are sort of nasty little bugs. They make us sick with the flu, but they make bacteria sick too. But remember, not all bacteria is bad too. And they have developed a method to protect themselves on it, and that's where CRISPR came out. And this is bacteria and archaea. Archaea is single-celled creatures that are there's chemical differences in the cell wall, and I don't want to get too much difference into it, but you could think of them as the same thing. But they're called prokaryotes. And now, what's a prokaryote? Anybody know? These are living things. They're single-celled. They have to be single-celled, and they have no nucleus. And there's a few other little things about them that make them different from the cells in our body. So we're talking about prokaryotes here, and I threw it in there just as a vocabulary lesson, and there's another one called eukaryotes that we'll get to a little later. So Ashino, in 1987, this goes back before CRISPR, discovered that what was later called CRISPR patterns in E. coli. He was studying E. coli bacteria. They live in your gut. And the bacteria had its own DNA, and it was not understood what these patterns were. There were strange patterns in the DNA. The big mystery is they called them CRISPR arrays. Why the CRISPR arrays were in the bacteria to begin with, and we'll see what they are in a moment. Now, this is a strand of DNA that's inside of the bacteria, but they just showed it as two parallel lines, and they didn't show the cross-hatching of all the ladder effect. So I just added that in right here. This is a schematic of a double-stranded DNA right over here. And what they discovered was that there were zones in it. Every other zone was identical. Those are the green ones. They were identical, and they were also read the same way forward and backwards. They were palindromic. which was very mysterious. So if you read the the nucleotides, the ladder structure, in one direction, then read it backwards in the other direction, it was a palindrome. You know what a palindrome is? Anybody give me an example of a palindrome in English? Lou, you must know. I forgot one. Level? Yeah, level reads the same way in both directions. So that's the idea here. But then between these palindromes, there were other sort of random zones, and he called those spacers. And they didn't know what those spacers were at that time. So the spacers were the colored ones, and they were all different, and they were in between. And this is what was in a typical strand of bacterial DNA. So the repeats, the pink ones, are identical and palindromes, and the spacers, all the other colors, appeared random and different from each other. And this was a big mystery at that time, and nobody knew anything about, what could this be? So in 1990 now, this is a few years later, bacterial DNA patterns got to be explained. And I repeated this little diagram over here. And it was Francisco Mojica. He worked with Jennifer Doudna, and he explained it. He said it was caused by viral attacks of the bacteria. So he shows a virus attacking an E. coli bacteria, and this is sort of what the bacteria looks like. The spaces between the palindromes contain sequences that were matched to particular viruses that were attacking the E. coli. So every time the bacteria got attacked, it stuck in another pattern, and a pattern was taken from the virus. And and if it survived the attack, it had a snapshot of the virus for the next attack, and it was able to identify that virus and take some defensive action. The E. coli stole an identifying DNA sequence from the virus and then inserted it into its own DNA. So it had a piece of the viral DNA in it. And the E. coli, they call these CRISPR patterns now, were immune to attacks from viruses. They had an identifying mugshot of the virus. They could tell when it was coming. Of course, it would have to survive the first attack. Other bacteria did the same thing. They said, well, what about, we're looking at E. coli. They looked at all kinds of other different types of bacteria, and they all had the same thing in it. The CRISPR allowed bacteria to develop an immune system, what they mapped this up into higher animals after. So how does the bacteria defend itself? And this is a sequence. You could think of these as E. coli bacteria. These little tiny things are viruses that are around them. And I'm going to go through the sequence pretty fast. The virus lands on it, shoots in its own viral DNA. A segment of the viral DNA is brought in and stuck into the bacterial DNA over here, and it added it in. And then a spacer was added. This was the spacer, and then another palindrome was added. And so this is how the CRISPR DNA built up in a bacteria. And these are repeated sequences over here. Those are the palindromes, and these are the things from all different types of bacteria. And then what the cell does, it converts that segment of DNA into an RNA molecule, just rips off one half of it. And this is called CRISPR RNA. And we're getting into the concept of CRISPR now. And then it breaks it up for each virus. Each virus has a little strand of that CRISPR RNA that's associated with the viral DNA. And then they pull in another, it's an RNA molecule called tracer. And I don't want to get deeply into that, but it's pulled in because it bonds with the CRISPR RNA, and it's able to now lock in the Cas9. You remember the Cas9 was mentioned, which is a protein that could cut up DNA. And so it gets into each one. And these are all for different viruses. And they now move along. They're going after the viral DNA. One of them locked on already over here, and this one too. And then the Cas9 acts like molecular scissors, and it will cut the DNA of the virus. That's what that's showing over here. And viruses don't have the ability to repair their DNA. Bacteria do, but not viruses. And we have it in ours. And it breaks it up into pieces, and I say we have a dead virus. Rest in peace. How does it work? In bacteria, this is sort of a review. The so-called CRISPR arrays in bacteria contain short samples of virus DNA called spacers that previously was attacking the virus. They were attacking viruses of the bacteria. The spacers were alternated by identical palindromic DNA code. I'm just giving a little bit of a review of what we just saw. And this is sort of a picture of what everything looked like that came in and attacked the virus. The spacer RNA that was cut out of the virus is over here, and then they hooked into this tracer that I talked about, and that locked into Cas9. These are the molecular scissors. So once we get, and they're going to search on this. This is the search argument. They're going to search the memory of the DNA that's in the virus, and when they get a match, it cuts it. So this is just a bigger picture of what we just saw right now. So this is the search argument. This is like searching memory. You could put a word and search your Word document, and it will find a hit. When it finds a hit, it cuts it. And these were two separate molecules. Now, how does it work? Again, now I'm going to introduce eukaryotes. We had prokaryotes. Anybody know what a eukaryote is? It's us. Higher animals that have nucleuses, and even pond organisms like a paramecium or an amoeba are eukaryotes. And so we want to see if we can get the same process to work on higher animals. And so what Doudna did is they hooked together this tracer RNA with the spacer that's doing the searching and made a single molecule that could go through and search any DNA. It could be in any organism, even a human. So this is a human-made invention. It's not merely a discovery of a natural phenomenon. By studying a phenomena in bacteria that took evolution over 1 billion years to perfect, they turned nature's miracle, that's what this was, into a tool for humans. And so here's the picture that we have right now. This is the Cas9 thing, that's the molecular scissors. There's the guide RNA that we're searching, and they could put in a search argument outside of the cell and bring it into the cell so that it would look for that. Might be an area where there's a disease sequence. And this is the thing that holds it in over here. And so here's a sort of a schematic of it. This came from Doudna's article. This is the Cas9 coming in and cutting the DNA sequence. And the cut is over here. At this point, it could be either repaired or modified, or we could bring in a new DNA sequence that was developed outside of the cell in a laboratory and stick it in there to replace a gene that was causing disease. And here, I'm just going to try this. There's your browser up to date. What do I do now? Do I say... I got that at home too.
Oh, what'd you do?
Maybe you could just try it over again. Okay. Okay, yeah, that was a good video though, but if I could, okay, that's okay. There's two terms in here that are used a lot: gene therapy versus gene editing. And gene therapy is a very slow process. It started before CRISPR, and it was engineered to counteract faulty genes causing diseases. And they were able to try to insert another gene. But gene editing is fixing the problem at its very source. That molecule that I showed you with the tracer in it and the argument that it's looking for could be put into the gene, find it, and fix it. With CRISPR-Cas9, we can edit or modify the genetic sequence found in a guide RNA. So Davna proved that CRISPR-Cas9 would work in human cells, and the human cells would now play the same role as the DNA in the attacking viruses. That was the big jump that she got the Nobel Prize for. And here's a little picture showing how that happened. It's a very simple-minded picture. But here's Cas9. There's the tracer that's holding it in. Whoops. And there's the thing that it's searching for, the CRISPR RNA. It found it. It cut it. And they either could delete it and bring it together, or they could bring in from outside in a laboratory another segment of DNA and stick it in. I keep touching it up. Let's see if this one will work. Gee, nothing is working. Okay. Huh. Yeah, I sort of, the videos actually are quite good. Because what I was planning to get into after this is the ethics. Let's see. Okay, go up. Well, that's one of them. If you can go up, I can get that one that we missed before. Is that it there? I think it is, isn't it? Yeah. Ah, same thing. None of the videos will work. Yeah. Okay. Okay, we'll see what happens. Okay, I think we talked about the differences between these two. Okay, there's something new. The leading edge in research right now is what they call prime editing. And they could get in now, when they search down, rather than pulling in another segment of DNA and inserting it where the Cas9 made the cut, it could actually go in there and nucleotide after nucleotide. A nucleotide is a rung of the ladder, make changes to fix a problem, like a disease problem. Normally, when they do the search, they might have to sequence the human genome to find out where like a disease gene is and go down and search for that. But when they get there, they could go in there and rung by rung of the ladder make changes. They call that prime editing, and that's the leading edge of research right now. So they could change individual characters like you would in a word processor. So CRISPR now and in the future, what will it do? Juicier tomatoes? No, the farmers like this. It's already being used to improve crops, GMOs. Make a coffee bean. There's a decaf coffee bean. Get rid of the caffeine right from the start. Make non-allergic allergic food, like milk. And produce a mosquito. Now, I don't know how it gets started, that only has male offspring. And they eventually die out. This I talked to some people about and wondered about the effect on the ecology. A lot of birds feed on mosquitoes. You know, if you could make the mosquito species disappear, what effect would it have on other things? This is what makes CRISPR very complicated and ethic-oriented, because you don't know what effect, when you're making such major changes that took evolution a billion years to do, and you go and change it like in a couple of weeks. bring them back to life. They're already talking about they can get DNA from mammoths, the woolly mammoth that's frozen in the ice, and by using an elephant, they could possibly produce the woolly mammoth again, or bring back the Neanderthal. The same thing through the same process. Or make faster racehorses. There's frivolous things we could use it for too. And one of the most interesting aspects of CRISPR is what could we do about aging? It turns out that we all have a gene that's the aging gene, and it's what causes aging. Even if you didn't get sick, or you try to live the most perfect life possible, you're going to get old. And there is a gene for aging, and it's caused. Aging is caused by aging genes in our DNA, or it's caused by gene damage too. From radiation and things like that could mess up your DNA. Both are fixable by CRISPR. Edit reproductive cells or embryos to pass it on to future generations. See later. And that is an extremely risky thing too. Live for 1,000 years? Theoretically, it's possible. What is the social impact? And what about the population of the world? That's sort of like the mosquito thing again. It turns out that the lobster doesn't have the aging gene. And I think that they'll just keep going on living until they're eaten up by another animal or humans, or they get killed in some type of accident. So there's an example. Maybe we should steal maybe borrow some lobster DNA. So therapy and ethics, there are two ways. In fact, I think I'm going to sit down and use this thing. I got a bad leg, so. Okay. We're going to talk about therapies and maybe the cure for diseases. And we consider somatic cell editing. Somatic means cells that are not part of the reproductive system. A muscle in your arm, the liver, your heart. You could get those cells and you could do some modifications of those in a lab and then reinsert them back in. And sickle cell anemia was one of those with blood cells. CRISPR could edit some, but not all, of body cells, somatic. So those are body cells. I'm using some buzzwords here just for education, but that somatic comes from the word body. I think it's Latin, and it could make changes that will not be inherited by future generations. And so we could take cells out of the body, edit them, return them to the body, and by delivering the CRISPR, or we could deliver the CRISPR tool into the cells inside of the body to begin with. We use this to treat blood diseases like sickle cell anemia. That was already done. So this is what happens in sickle cell anemia. These are the blood cells that are shaped like a sickle, and this was cured. And these are some of the other diseases that potentially could be cured by CRISPR. And one of the big ones here is cancer. Cancer is a genetic disease, and the T cells could be modified in our blood to attack cancer cells. And that's what this is trying to say right here.
Can I ask you a question? Do you think this would be more preventative or?
This one would be or whenever cancer cells start up. I'm not too sure. In fact, this thing just quit on me. I better stand up again. Okay, so I wanted to get into some of the ethics of it. The ethics is a real complicated area. Germline editing, we just did somatic cell editing. Germline editing means modifying the DNA in the reproductive cells of a person, an animal, or embryo, an embryo that only has a few cells in it. So these two pictures are showing that here. So CRISPR editing of the DNA in a human egg or sperm or early embryos could result in that the children will now pass on the traits from generation to generation, and we changed our evolution. This has the potential to alter our species and control our evolution. National and international science and medical agencies limit and prohibit germline editing. It's in the U.S., it's just there's a lot of pressure against it, but there's no laws or anything like that. And there's something strange that happened in China. Francis Collins, who used to run the NIH, said NIH will never fund any use of gene editing technologies in human embryos. So this one I think I could do.
A for-profit biotech called Bluebird Bio developed a gene therapy treatment for sickle cell disease, and they were going to charge $2 million for it. They were unable to get European governments to pay that much, and rather than take a price cut, they just left the European market. And recently, we were all sad to find out that the company is about to go potentially out of business. Making money on therapies for such rare diseases is not simple. And so, folks, the situation is dire, and the issue is stark. Rare genetic disease is not rare. What is rare are cures for them. And when these cures exist for about 10 out of 5,000, they're 2 million a patient, and the rest are, quote, not commercially viable. I would not be standing here if we did not have a path forward. And the solution that we aim to bring here is gene editing with CRISPR. So gene therapy that I told you about, using a virus to put a gene back, really works, but it is expensive and can be unpredictable. So by way of an analogy, gene therapy is like having a car with a flat tire and adding another wheel. Gene editing is repairing the flat. It's like having a word processor for your DNA. And the engine that powers gene editing is this wonderful molecule, CRISPR. A for-profit biotech called Bluebird Bio.
Okay, I got it over here. This is another area that to me was one of the biggest areas of ethics in this thing. It was what I call designer babies. Genetic engineering will change everything forever via CRISPR. It's already happening. Although China had regulations against germline editing, Zhui, I'm not good at pronouncing his name, edited the genes in a viable human embryo to create genetically modified babies. The twins were born. The intent was to protect the babies from HIV virus. He changed the DNA so that they would not ever catch HIV or AIDS. Their parents had AIDS already, but the twins were born and they would be immune to AIDS. All future offspring would inherit this trait. Sounds like a good thing, maybe, huh? Zhui was soundly criticized and reprimanded by the scientific community. Jennifer Doudna was very much against this. This happened only in some, 8 years ago or so. And these are pictures of the babies, very cute. And his designer babies were here. They were immune to AIDS. But you know what the Chinese government did? He was sentenced for 3 years in jail for doing this. So what's the future? What if the next CRISPR baby is edited to have desirable physical characteristics, such as high intelligence, super athletic ability, be a warrior? We now have the ability to alter our own species and control evolution. What if this gets into the hands of some country that's would be considered an enemy and wanting to take over the world, like in World War II with Hitler. So will this be a manifestation of the prophetic book? I read this book in college, The Brave New World by Aldous Huxley. Did anybody ever read that? It gets into these types of ethics. And in this novel, human beings are genetically engineered. Yeah. So this is what it reminded me of. The government in this novel manufactures babies. I put that in quotes. They're modified to belong, and I think I have something that's missing off of there, to a certain group that they came up with. So this is my last slide. And the Nobel Prize, of course, was in just a few, 5 years ago. 5 years ago. Jennifer Doudna, Emmanuel Charpentier, they won the Nobel Prize for their work on CRISPR. And the Secretary General of the Royal Swedish Academy, who gives out these prizes, says this year's prize is about rewriting the code of life. Now, that is the end of my talk, and I'm open to any kind of questions or comments that you might have. What about the ethics of it? Oh, yeah.
Okay.
Oh, yes.
Try to go into the PowerPoint and copy the URL in this page.
Oh, yeah, those two yeah, could we do that? Yeah.
You can do that, but do you have any questions right now? I know there was one question from the chat.
Okay.
Let me get to. And it says, can we use CRISPR to cut out shortened telomeres and extend longer telomeres to essentially extend human life?
That's what that one slide is that I had.
Yeah.
There is genes in there that deal with that, and they talked about the theoretical possibility of living forever. I mean, unless you got killed on something or run over by a car or whatever, you know. Yeah, okay. Oh, yeah, that's what we got to do. Was there one before that? That was the first one, right? Yeah.
All right, so
Yeah, good point.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences.
That's when it's
It
cut.
effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
Is there?
Hey guys, welcome to my new
Here's another
video.
one
In
there.
this tutorial,
Okay. Here's the escape key. And that's down below that, further down. Wait, no. Is that it? Oh, let's show that one there. Is there a video for that? No. Maybe it's up above that one that we just looked at. I think it is. We just did that one. And, okay. Maybe, is it that one? Yeah, I think that's what the one, yeah, I think that one.
That's the one you want.
Yeah. That one wouldn't work, I remember. Here's the other video that we missed. Oh, did we see that? Did we see this one?
Yeah.
Oh, okay, I'm sorry. I don't know where it is then.
All right.
Yeah.
Is there any other questions here locally?
Yeah.
All right.
Okay, I wanted to show you something. A couple of summers ago, I taught a summer class in Search for Life Beyond the Earth, and we did get into DNA, and that's what sort of got me first interested in this stuff. And we were wondering about, whether there could be DNA on other planets or anything like that. And I had the students actually make a DNA model. And I have a couple of them up here. I made some before I came in, and I'm going to get over there. What I did, it turns out that there's an awful lot of computer science in DNA stuff, because it's a memory, and you can encode it. And I have right here from Twizzlers, and I took my name and encoded it with the base pairs. The ladder here has two different molecules, A, C, T's, and G's hooked together. Now you can't hook an A with a G, but I did it anyway. I cheated here to make the thing short. And what I did is I took the list of all the letters from A to Z and looked at their. You know what an ASCII code is? It's the codes that are for the keyboard of your computer. And they're numbers, some 97, 98, 99. And I converted them to a base 4 number, and then associated the colors of the ACs and Gs and Ts to those numbers, and then mapped those into colors of straws. I have 4 different straws of 4 different colors, and I encoded my name, which is Nick, in this piece of DNA, and it's in a Twizzler thing here. And you could actually eat the Twizzler when it's all done. I ate about 3 or 4 of them while I was making this thing here. I have another one here. I unraveled it, but this is my name in these 2 right over here. So we had students do that. They actually encoded their names. It had to be a pretty short name. And you could put a restriction on that you cannot hook together like an A with a G. But I dropped that restriction just to make it easy. But then it takes more of those rungs to encode one single letter. It takes 2 rungs for a letter. This would take 4 rungs for a letter now with that restriction in it. And so that's what we did on that thing here. And what I have here, for anybody who's interested, for a science fair, I have an instruction sheet that shows how to get the soda straws cut so that you could put your name in it, and a picture of a partially finished DNA model that I made. And I have them right over here. There's a bunch of soda straws. There's a bunch of Twizzlers. They could either eat them or try to make a DNA model out of them, and a bunch of toothpicks. And if anybody's interested in this, you're welcome to come up and take it. If you have students at home that might want to use it for a science fair, I think it would make a good science fair project. You're welcome to come up and take more than one of everything, because the Twizzlers break while you're puncturing them. It was pretty tricky figuring out how to do that, but that's the challenge of it, So, but anyway, anybody here have any comments on? I was wondering about the ethics of this stuff. I mean, where is this leading us? And how am I doing on time? Pretty bad. The ethics of doing germline editing. That's what we were said. Which we could control our own evolution, and are already changing the evolution of animals to have cows that produce more milk and eat less grass or something like that. And there's or like the mosquito thing I saw. So it's already being done in animals. And what about humans? What if this idea got into the hands of, I hate to say it, but North Koreans who don't like us too much, like in the Brave New World, could somebody try to produce a super army? And or make people that all they want to do is menial work. They'll be our workers and leaders and all the different types of people, various colors of skin, you know. So that was one of the big points that I sort of wanted to emphasize here. And I guess if there are no more questions, I'm done.
All right. Now, there weren't any comment on the chat was that ethics-wise, if we don't make use of technology, some other country would, i.e. atomic bomb technology in World War II.
Yeah.
And if there's not a fear of abnormal development of a life form, and so the need to abort, it would welcome the unextinction of the woolly mammoth as long as it was not physically and mentally harmed. I guess that's the question is what we don't know.
Yeah, well, a lot of those creatures that got extinct, they were for a reason. You know, all through the history of life on Earth, animals came and went, you know, and you bring them back again now. Is that a good thing? I don't know. But think about the ethics of this. If you had the ability to cure cancer, and you wanted to put it in, and you wanted to make it into the genes of embryo, so that all of its offsprings would never have cancer again. It sounds like that would be if you didn't do it, that might be considered immoral or unethical. But if you did do it, all of a sudden you're controlling evolution again. How do you know what side effects it's going to have? Maybe some of those genes have some subtle side effects on other things too.
Also, in your presentation, it was $2 million.
Yeah, that one.
It was $2 million a person.
That one video, yeah.
So it seems like to me it's going to be a big.
Yeah,
big
that's a
event.
piece
The
of
people
it.
that have it, have
Yeah.
the funds to do it. Their offspring won't have cancer, but if you can't afford the the millions of dollar gene therapy or the gene editing, your offspring and you are stuck with the diseases that we still have. So
the video that mentioned that he was very much disturbed by the idea that money now could stop the cure of diseases. You know.
well, it does now.
It does now too, yeah. So
Because if you're not paying someone to work, they're not
Yeah.
going to go to work. Like and I don't blame them. I mean, no one's going to do this for free. Like no one's going to have like a free career. Like how are you going to pay your electric bill? So it's nice to say it should all be free, but how? What doctor is going to show up to do that with no reward, no financial reward of any kind?
Yeah.
So it's
maybe it's where the government steps in. I don't know.
All right, well, we could certainly continue this discussion if you like, but I also want to mention that especially for those people that did come and pay, you have a rule here that you can't leave Kopernik until you look through a telescope, and unfortunately tonight's not the night. So if you did pay, you can go back to the cash register and get a card to come back on a subsequent night for free. So thank you, Dr. Guydosh.
Okay, you're welcome. Thank you very much. Thank you very much.
So next Friday's talk is about biofilms, and then the Friday after that is black holes on Black Friday. Hope to see you there.
If anybody wants any of these things for making models, feel free to come up and take them. I'll leave them here for a while.